Good morning, everybody. On behalf of the US FDA and the Center for Research in Complex Generics, I would like to welcome you to this workshop on regulatory utility of mechanistic modeling to support alternative bioequivalence approaches. For the next 10 minutes, I will give some introductory remarks to introduce a Center for Complex Generics and as well as discuss some logistics of the current workshop. So here are two topics. The Center for Research in Complex Generics was created about a year ago by the award of U18 grant by the US FDA to two universities, the University of Maryland in Baltimore and University of Michigan. It is co-directed by James Polly at University of Michigan and myself at University of Maryland and myself at University of Michigan. And the center manager is located at the University of Michigan, Visha Krishnan. We have an email. You could please subscribe to the listserv, and we post all of our events information on our website, www.complexgenerics.org. All the information about this current workshop could be found there. The mission of the center is threefold. We have really three main objectives. The overall mission is to increase access to safe and effective generic drugs through enhanced infrastructure, communication, education, and research collaboration across industry, academia, and FDA. And to accomplish that, we have three main goals. So the first one is to communication. We would like to establish this independent route of communication between the stakeholders, such as FDA, industry, through the center to assure success and to assure that we have a better understanding about the actual problems generic industry is facing. The second mission is education, is to promote those kind of training workshops and seminars, as well as hands-on demonstration, and to engage the students and scientists at the FDA and the company in this complex generic research. And the third mission is research, is to initiate some small collaborative projects between the center, FDA, and industry to facilitate answering the questions that are hampering development of complex generic drugs. So, as you know, communication is a central mission of. Sorry. So communication is our central mission. I guess I didn't delete the previous sentence. And because of this communication mission, what we are doing is we are reaching out to industry to ask them what are the main questions and issues they're experiencing. And to do that, we have performed two different activities. One was to initiate survey on the scientific challenges in development of complex generics. So survey was conducted and many generic companies and scientist representatives have answered the questions on the survey, and we will be publishing the results of the survey very soon in the APS journal. So in the survey, we asked three questions. First is which complex products are the most important for the companies, and we give all those possibilities: complex injectables, complex mixtures and peptide drug device combinations, inhalation and nasal products, long-acting injectables and implants, ophthalmic products, dermatological products, and then other drug and drug products. And then we ask what kind of analytical technique or scientific techniques are the most important with respect to those products, and which educational topics are the most important. So based on the survey results, the results came in and there was many topics were highlighted, and among complex products, complex injectables, formulations and nanomaterials, then drug device combinations and nasal and inhalation products, but others were mentioned as well, transdermal and ophthalmic. And if you look here around analytical techniques, what were the most interesting comments that people wanted to have some access to locally acting physiologically based pharmacokinetic modeling, oral absorption models and bioequivalence, data analytics and machine learning. And around educational topics, again, there is a big interest in data analytics, including quantitative methods, modeling, and simulations. So because of that, we really decided to conduct this workshop that we are here together to get some access and to understand the status quo of the modeling in the field. So in the interviews, we also interview about 20 different organizations, some of them multiple times. And we interview both large generic companies, small specialty pharma, and also trade organizations. And in the interviews, again, the number of products came up over and over again, and these were complex injectables, inhalation, transdermal, ophthalmic, orals. And among those most important issues, again, as you can see, bioequivalence issues, clinical trial design, use of modelings, were very prominent questions brought up by the generic industry. So I kind of tried to summarize in my GADUFA presentation in May, what were the main issues brought up by the modeling and what is the most important topics around the modeling in generic industry. And they pretty much range around this PBPK for inhalation products and solid oral dosage forms. Very large topic is long-acting injectables, in silico modeling for ophthalmic topical and inhalations, and then populational PK with the site of absorption for the, again, inhalation products. And when generic industry brought up concerns, they were pretty much like that. It's very important to find alternatives for the design of complex and expensive endpoint clinical trials, but generic industry doesn't want to be trailblazers. There is a kind of a lack of universally accepted models for generic drugs. There is a lack of publication on the expectation for bioequivalence modeling and requirements for the validation of the modeling, very broadly brought up item. And then all the companies would like to have a feedback on the models early in their development. And then they're not sure what will be accepted and how it will be reviewed. And then can this modeling supplement some of the patient data if it's missing? So with this workshop, we are setting up as very interactive. So we really would like for the industry representatives and whoever call in the discussion sections to put your questions in the chat box and address them at specific speakers to make sure that some of those items are addressed. And as the center grows and as we have some in-person workshops, we really hope to have more inclusive environment where the companies and the FDA and the scientific leaders will have opportunity to exchange ideas and ask and answer actual questions. So the training mission of the center, again, is involved around creating workshops and also webinars, but also with the end of the pandemic, we hope to have some hands-on demonstration and some generic Scala program. So the workshop is here. We performed so far one workshop in August, IVRT. And for this workshop, currently you could find the slides posted at the CRCG website, and soon the recordings of the presentations will be available. Today, we are starting this second workshop on regulatory utility of mechanistic modeling. And it runs today and tomorrow. And again, you could find all the information on our website. And the third workshop planned for this year will be conducted on November 30th. And it's focused specifically on establishing modeling for long-acting injectable products to demonstrate bioequivalence of them. So please register for that one. We also have a research mission, and in this research mission, we initiated one of this important project around long-acting injectable modeling and designing of the models for the bioequivalency alternative. So here is our workshop, and you will have several other people discussing what's going to be happening. So today we will discuss modeling of orally inhalable products from 9 to 12, and then dermal products between 1 and 3, and then other locally acting generic products 3 to 5. Please make sure to call in at the very last moment, at the last hour when this panel discussions are happening, and please make sure to put in the questions. The agenda is available at the website. The biographies of the speakers are also available, and we are allowing ample time for discussions. So go ahead. This is our way to have this more interactive. We plan to so we will have a mixture of recorded talks and this live panel discussions. You have a URL for each of the day. Please enter questions in the chat, and if you want to direct your questions at the specific speaker, put it at and the name of the speaker. We plan to put the copies of the all the recordings for this workshop around 3 to 4 weeks past the workshop and our website and complex generic products. With that, thank you very much. Thank you for registering. Thank you for participating. And yes, to participate, you have to ask those questions. Many thanks to the speakers, moderators, panelists, but also to the organizers for this workshop, especially from the FDA side. Andrew Babinski, Lucy Fan, Feng Wu, Lei Zhang, Lian Zhao, and then Sam Rainey. And a special thanks to Dana Hamill from University of Maryland and Visha Krishnan from University of Michigan for making sure this event goes smoothly. So with that, I would like to introduce the next speaker. And yeah, the next speaker is Dr. Jaqueline Corrigan-Curay. Dr. Jaqueline Corrigan-Curay is currently the Principal Deputy Center Director at the FDA Center for Drug Evaluation and Research, CDER. And then most recently, she served as the Acting Center Deputy Director of Operations, leading center and agency level priority and initiative programs, and especially leading GDUFA 3 reauthorization negotiation. Dr. Corrigan-Curay earned her JD degree from Harvard Law School and her medical degree from University of Maryland School of Medicine and bachelor from Harvard. She has been active participant of many panels and many initiatives within the government agencies, including FDA, NIH, and other regulatory bodies. Thank you.
Good morning, and thank you for inviting me to open this very exciting day, and to the Center for Research on Complex Generics for co-hosting this great event. It is so impressive, the breadth and depth of expertise that has been brought together today to work to advance our understanding of mechanistic modeling. The FDA that established the Center for Research on Complex Generics is an example of how enhancing research collaborations is increasing access to safe and effective generic products. Advancement of generic drug science is a collaborative process. We appreciate these opportunities to collaborate with the generic industry and its stakeholders through exchanging research, training, and resources. Regulatory decision-making must be based on the most current scientific evidence. We do that by ensuring that the methods needed to establish bioequivalence are grounded in the most up-to-date science, are as efficient as possible, and that the approaches to develop generic drugs and to assess abbreviated new drug applications use 21st century technologies. Modeling and simulation have been increasingly used in drug development and regulatory review, providing a framework for synthesizing information, streamlining clinical trial design, enhancing individualized dosing, and extrapolating beyond what has been studied. Simply put, the goal is to close the gap between scientific innovation and drug review. Under PDUFA 6, the Model-Informed Drug Development Pilot Program was launched with the aim of facilitating the development and application of exposure-based biological and statistical models derived from preclinical and clinical data sources. The Model-Informed Drug Development approaches use a variety of quantitative methods to promote better understanding of the risks and benefits of drug products in development and to improve the efficiency of drug development. When successfully applied, model-informed drug development approaches can improve clinical trial efficiency, increase the probability of regulatory success, and optimize dosing therapeutic individualization in the absence of dedicated trials. Ultimately, it is the right drug at the right dose for the right patient. Model-informed drug development applications include predicting clinical outcomes, informing clinical trial designs and efficiency, supporting evidence for efficacy, optimizing drug dosing, therapeutic individualization, predicting product safety, and evaluating potential adverse event mechanisms. These innovations inform drug development policy. Modeling and simulation approaches have been widely used to support new drug development under the IND stage and regulatory decisions under the NDA and BLA stage. During the pandemic, modeling and simulation approaches have been used to identify potential candidate compounds for treating COVID-19, to extrapolate efficacy findings into untested populations, for example, pediatrics, and to select dosing regimens. Science often moves quickly, and we need to be as nimble. An example of this is the Fit for Purpose initiative, which provides a pathway for regulatory acceptance of dynamic tools for use in drug development. This program recognizes that science may evolve for certain types of drug development tools, and the formal qualification process may not be the path for everything. Therefore, again, making sure that our regulatory policy is responsive to science, a designation of Fit for Purpose has been established. The importance of generic drugs to Americans, and indeed the world, cannot be overstated. Moreover, the need to approve complex generic products is a particular priority. GDUFA-supported research has helped develop core modeling and simulation tools for generic drug applications. GDUFA-supported research has yielded better models for key locally acting routes of delivery, topical, ophthalmic, inhalation, and nasal, to the community. This workshop will cover the next step, applying these models to regulatory decisions, specifically the high value of using models to support and evaluate more efficient bioequivalence methods. Quantitative methods and modeling support better and faster decisions during the generic drug lifecycle because they integrate knowledge accumulated during and after new drug product development. This includes in vitro in vivo correlation for formulation design, absorption, distribution, metabolism, and excretion properties. Indeed, quantitative methods and modeling allow the Office of Generic Drugs to rapidly respond to new regulatory challenges that arose due to the COVID-19 pandemic, such as disruptions in vivo bioequivalence studies and interruptions to the supply chain of the proposed generic and reference products. Continued focus on innovative approaches such as adaptive designs and model-integrated evidence generation will enable acceleration of access to generic drugs through increased efficiency in drug development. Of course, drug development is a partnership. Public workshops like the many that FDA and the Center for Research on Complex Generics are co-hosting over the coming months provide an opportunity for a focused dialogue between the FDA and the pharmaceutical industry, including supporting industries and other key stakeholders on how enhanced regulatory decision-making can be realized from an emerging tool or technology. The tool being discussed today is mechanistic modeling. While it's been around for decades, its usage in generic drug development and regulatory assessment has to date been limited, particularly from the applicant side. As the field continues to grow and advance, our goal for workshops like this one is to chart a course to expand integration of mechanistic modeling into product development and identify best practices to accelerate drug development time and optimally move to approval more rapidly with fewer cycles. At the workshop today and tomorrow, you will hear from leading experts from the FDA, generic and new drug industry, academia, and other research organizations. and developers of the modeling tools on the use of mechanistic modeling, such as physiologically based pharmacokinetic modeling to support alternative bioequivalence approaches for complex, locally acting products and orally administered products. I want to thank you all for being here today, for your attention, and without any further ado, I want to turn you over to get started on the science. I wish you all a very productive workshop.
So our next speaker is Dr. Shiew-Mei Huang. Dr. Huang is currently a deputy director in the Office of Clinical Pharmacology for Center for Drug Evaluation and Research at the FDA. She received her bachelor degree in pharmacy from National Taiwan University School of Pharmacy in 1975 and her PhD degree from the University of Illinois in Medical Center in Pharmacokinetics and Biopharmaceutics. Following her graduation, she worked for over 15 years in the pharmaceutical companies, Ortho Pharmaceutics and DuPont Merck, and joined FDA in 1996. She co-authors over 160 publications on various topics in clinical pharmacology, drug metabolism, and physiologically based pharmacokinetics modeling. Dr. Huang is a fellow of AAPS. She also was the president for the American Society for Clinical Pharmacology and Therapeutics in 2009 and 2010. And she received multiple awards from the same society, such as Gary Neal Prize for Innovation in Drug Development and Henry Elliott Distinguished Service Award. Welcome, Dr. Huang.
Good morning. I'm Shiew-Mei Huang from the Office of Clinical Pharmacology, FDA. In this two-day workshop, we will hear about the progress and challenges in the use of mechanistic modeling and simulation, including PBPK, in addressing complex issues in generic drug development and review. What I would like to do is to provide a clinical pharmacology perspective on the use of PBPK in new drug development and regulatory review. The MIDD model-informed drug development is a development and application of exposure-based biological and statistical models derived from preclinical and clinical sources. MIDD has been increasingly used in both drug development and regulatory review. It informs study design, the evaluation of various intrinsic and extrinsic factors on exposure and response. And PBPK is one of the key components. We look at the PBPK application submitted to the Office of Clinical Pharmacology. Looking at the left, and in the 10 years ending in 2017, we average about 25 to 30 applications per year. And the majority is on drug-drug interactions, that's enzyme-based or transporter-based, or in the application in pediatric application. In the last 3.5 years, again, we see the majority, more than 2/3, in drug-drug interaction that's enzyme or transporter-based in pediatrics, and with increasing use in other areas. So in our office, the PBPK new drug application is managed by the PBPK team in the Division of Pharmacometrics. The co-leads are Susie Zhang and Yuqin Yang, and with members that we have recruited from industry and academia. So the roles and responsibility of the PBPK team was to develop best practices, guidance on the use of PBPK, and the important day-to-day work as regulatory review to coordinate, conduct PBPK reviews. And in the past, we have done de novo model development that recently the majority time is to evaluate submitted PBPK models and ask for refinement if needed, and maintain a PBPK knowledge base, and then to provide the experience that we gain from our review and additional research with various groups, with internal and external experts. The team also organizes or attends outreach programs, workshops, scientific conferences, and they harmonize with other health regulatory agencies. For example, Project Orbis, we conduct simultaneous review for applications submitted to the US, Canada, Australia, Singapore, Switzerland, and the UK, and to also foster communications with thought leaders, for example, with sabbatical program and others. So for PBPK review, at the IND stage, it's to look at the application of PBPK analysis. As indicated earlier, the majority is to support the DDI evaluation to inform protocol design, or to support initial dose selection in specific populations, and or to support dosing under emergency use authorization. And the discussion focus is mostly on the acceptability of the proposed modeling approach. The PBPK review focus at the NDA stage is to the following. Here we have a brief chart. First, to identify the model impact, to identify the context of use, and look at the labeling claim and the overall model risk, to look at the impact on patient care. And then to review whether the model is adequate. Here, review the mechanistic assumptions, looking at the in vitro review data, and if the model is deemed inadequate, then the PBPK data will not be used in the labeling, and there may be post-marketing commitment or requirement studies. If it's a yes, there perhaps are additional simulations to look at alternate dosing regimens or alternate mechanisms, and the information can be included in the labeling. And when we think the model is not adequate, the model can also be refined and follow the green chart. So the challenges here at the NDA stage, as also will be discussed later, is some of the uncertainty in system parameters and also limited confidence in vitro in vivo extrapolation. Also, whether we have appropriate model assumptions, and internally for regulatory review, sometimes the short review timeline is a challenge. So here is a display of the regulatory application and predicted performance based on our early applications. Here are three big areas: drug interactions, specific population, and other areas. And on the upper left, there are color-coded areas to show our confidence in each area, with green the most confident, and then yellow with some experience, and red as limited experience. And you can see with drug-drug interaction, we have a lot of experience with enzyme-based interaction and increasing understanding of the use of and the with the transporter-based interactions for specific populations and other areas such as food formulation effect, pH effect, absorption. There are increasing applications, and today we're going to hear more about the progress and challenges in these later applications. Next, I'm going to use three cases to show the successful applications of PBPK in new drug review. So the first case is drug-drug interaction. This siponimod is a CYP3A and 2C9 substrate, and it was approved as an oral product for multiple sclerosis 2 years ago. And here the PBPK model, when it's developed, it look at the information about its absorption, distribution, and elimination. And the model is validated with the available single dose, multiple dose, and PK in various genotypes. Also, the in vivo drug-drug interaction data with rifampin and fluconazole in certain genotypes. So the application is to evaluate the combined effects of 3A, 2C9 modulators on PK in various 2C9 genotypes. Here I highlighted those that we don't have information, and those are predicted based on the model. So it has informed the labeling about various genotypes of 2C9. For example, star 1, star 1, star 1, star 2, and star 2, star 2 is 2 milligram. And here, star 2, star 2 in red are where we don't have data, and this is based on prediction. And then star 1, star 3, star 2, star 3 is 1 milligram dose. And star 3, it's contraindicated. And then in addition, there are information on the use of siponimod with either moderate 2C9 and moderate or strong 3A inhibitors when they're not recommended. Here I only listed those that's not recommended. So it's a very complex situation based on PBPK modeling. So the second case, it's about baricitinib. So baricitinib was approved in 2018 for moderately or severely active rheumatoid arthritis. And later, it was authorized, for example, in November 2020 for COVID-19 to be used together with remdesivir. 2 months ago in July 2021, it's authorized to use it by itself. Does not need to be used with remdesivir. So here, the context of use is that clinical data showed that its co-administration with probenecid increased the exposure of baricitinib by 2-fold. So what can we expect when we co-administer baricitinib with a weaker O3 inhibitor such as ibuprofen? And if you look at the review, it indicated that 90% of the baricitinib absorbed into the body, renally excreted, with active secretion accounted for 50% of the renal clearance. So the sponsor developed the PBPK model with mechanistic kidney transport pathway. So here you look at pemetrexed, which is O3, O4 substrate, and baricitinib, O3 and Mei substrate. And you look at the ibuprofen and probenecid, the in vitro inhibition data are obtained. And then the sponsor verified the PBPK model with clinical PK with DDI studies. So there are available pemetrexed with ibuprofen, and then also baricitinib with probenecid. And then with those data to predict the substrate, either pemetrexed or baricitinib, the exposure under untested drug-drug interaction scenario. So here it's either with diclofenac or ibuprofen. So the PBPK modeling enabled us to predict the minimal effect of ibuprofen, for example, here a weaker O3 inhibitor on baricitinib. So if you look at the labeling, it says the usual recommended dose is 2 milligrams once daily. However, if you're taking a straw or getting an anion transporter, the O3 inhibitors such as probenecid It's 1 milligram once daily. However, based on simulation, PBPK simulation, with diclofenac and ibuprofen, which are O3 inhibitors with less inhibition potential, it predicted minimal effect on the PK of baricitinib. And in the labeling, there is a forest plot to show that with probenecid, there's more than a twofold increase, and so recommended dose is 1 milligram once daily. With ibuprofen, diclofenac, the AUC and Cmax didn't change much, so there was no dose adjustment. The third case is again on baricitinib, is to predict the PK in pediatrics. Since there are no PK data in adult patients with COVID-19, and 4 milligram QD dose was recommended based on the dose used in the platform study. ACT2, ACTT2, the adaptive COVID-19 treatment trial 2. So without the PK data, what should be the dose for a pediatric patient with COVID-19? So the review look at the various from various data sources, look at PK in healthy adults, PK in adult patients, and also to look at PK in pediatric patients with different diseases. So the assumptions in the modeling is that the effective exposure in pediatric patients 2 to 18 years is the same in adult patients, both with COVID-19. And the PK difference between adults and pediatric is similar across different diseases as listed above. So the baricitinib, its emergency use authorization was again based on the dose for adults was based on the clinical efficacy study, and the dose for pediatric was based on the analysis of historic PK data I showed earlier, and the PBPK modeling, which I didn't show the detail. But the modeling showed the information for dosing. For example, 4 milligram for adults and 9 to 18 years old, and 2 milligram for 2 to 9 years old. And then based on the renal function, the eGFR, the doses were changed accordingly. So these are the 3 cases where PBPK was applied successfully in the for regulatory decision making, and also its impact on the labeling. So this paper, published in November 2020, it has an update on submissions to the to our Office of Clinical Pharmacology, and it has a large table summarizing a recent regulatory review and labeling. So, for example, it may show that we need post-marketing required studies and post-marketing commitment studies, and there were not information in the labeling on the PBPK. And at certain situations, we may think it's adequate, and we have the labeling information based on modeling in the clinical pharmacology or drug-drug interaction section. And there are also cases where we consider the PBPK modeling application is inadequate, and with the reasons written in the table and in this paper. So it is critical that we need to develop the best practices in physiological-based modeling, and this is a workshop that we held in November 2019, and the paper was published in CPT PSP. So in this paper, it summarized the highlights of the workshop and also extensive discussion summarizing the challenges in the application of PBPK in 3 areas. We need to increase the effectiveness and efficiency of regulatory review. We need to increase data acquisition, analysis, and transparency, sharing of data, and to harmonize the PBPK credibility criteria in drug development and regulatory submission. And there's a big table about the challenges and recommended actions. So in summary, PBPK is a critical component of MIDD with increasing frequency of use. In new drug development, the applications have been in the drug-drug interaction, either metabolism or transport-based, in pediatrics, and in other patient factor evaluations, such as in renal or hepatic impaired patients. The challenges need to continue to be addressed. Here are just a few. I mentioned earlier uncertainty in some system parameters, and here are just some examples. The ontogeny, the interleukin levels, and specific population information, and limited confidence in vitro in vivo extrapolations, especially concentration in the tissue, and validity of some of the model assumptions. Here internally, the regulatory application, the challenge is a short review timeline, and there are many others as stated in our publication. And then it is critical to extend the applications to integrate the effects from multiple patient factors, such as pediatrics and drug-drug interactions, or in older adults and drug-drug interactions, and renal impairment and drug-drug interaction, or genetics and drug-drug interaction. I did have an example of that, and the food and pH effect and others. We need to continue to establish good practices in PBPK modeling of both new drug and generic drug application, as stated in slides number 8 and 16. Finally, I'd like to acknowledge individuals for the PBPK review. Here's Office of Clinical Pharmacology, PBPK team, our review staff. And for today's slide preparation and review, Susie, Eugene, Jiang Huang, Ken Chi, Kelly, Shening, Liang, and Isam, and also the IND, NDA, BLA applicants who have provided the PBPK models and academia collaborators. Thank you for your attention.
So I'm happy to introduce the next speaker. It is Dr. Andrew Babiskin. Andrew is currently holds the position of team leader for locally acting physiologically based pharmacokinetic modeling team in the division of quantitative methods and modeling in Office of Research and Standards and Office of Generic Drugs in CDER. His work is focused on establishing of the novel in vitro and in vivo pharmacokinetic-based methods to establish bioequivalence in lieu of bioequivalence study with clinical endpoints, so very important for generic industry. Andrew was very critical in setting up this current workshop. He was a leader of this organization team for this workshop, so I really would like to acknowledge him in that role. Andrew received his bachelor degree from University of Maryland College Park in chemical engineering, and his PhD degree in chemical engineering as well from Caltech. And he's been working at the FDA since 2014. Welcome, Andrew.
Sorry, I was on mute. Thank you, Anna. Good morning, everyone. I want to first take the opportunity to thank everyone for attending today. But I also want to thank the FDA Organizing Committee and all the CRCG organizers in bringing this event together. I also want to thank all the presenters, moderators, and panelists for the significant amount of effort they put forth to make this workshop happen, including our multiple planning meetings. Today, I'm just going to give you an overview of what's going to happen today. Just a position where we are right now. You just heard the opening remarks, both from CRCG and also from Dr. Corrigan-Curay. We also just listened to the keynote speech from Shiew-Mei Huang, the Deputy Director of OCP. Now we're getting into Symposium 1, which is mechanistic modeling of locally acting drug products. There's going to be 3 sessions that focus on early inhaled, dermal, and other such as ophthalmic and nasal locally acting drug products. This symposium really correlates with GDUFA efforts to expand access to complex generics as mechanistic modeling can be utilized to support bioequivalence approaches, but also to address complex review issues related to bioequivalence. Each session will include experts from the regulatory and non-regulatory side, such as from the generic drug industry and model developers, including academia. In each session, you will hear a series of pre-recorded presentations. Note that the presentations in this session will be played in succession, so the times that are listed in the agenda are only ballpark estimates of when the presentation will begin. After the presentations have completed, we will then enter the live panel discussion, which will include the speakers and then additional panelists. Each panel will also have 2 moderators, one from the regulatory side and one from the non-regulatory side. Prior to each session, the introductions will be provided for the speakers and the moderators, but when the panel goes live, you will then hear introductions for the remaining panelists. Please refer to the biographies document that's on the CRCG website for this event for additional information on the participants in today's workshop. And as previously mentioned, the presentations and panel discussions will be available later after the workshop has completed. And also to reiterate a point from Anna from the opening remarks is that we really encourage everyone to add comments and questions into the chat box. This can be done even while presentations are being played or during the live panel. If relevant to discussion, the panel will make the attempt to incorporate these comments into the panel discussion. But if not, we really appreciate this input because after the workshop, we'll be able to collect together both the FDA and CRCG and discuss priorities going forward. So with that, we will enter the first session.